Okay, welcome. So for those of you new to Taste of Science, welcome. Welcome to those of you who are not new to Taste of Science. So we're a part of a national nonprofit science outreach organization for adults. And usually we'd be holding a big in-person science festival in cities all across the US, inviting you to meet your friendly neighborhood scientists in your favorite spaces, bars, and things like that. And we really hope that we're going to be back to that soon. But for now, we have several virtual events planned throughout this week. And there are actually a couple in-person events as well. You can check out the schedule at tasteofscience.org. So this event is being put on by our New York City chapter. I'm Catherine Hoffler. I'm a volunteer for the New York chapter of Taste of Science. And tonight's event is about how computers and artificial intelligence are helping scientists learn about protein structure, design better drugs, and generally do better science. So before I introduce our first speaker, I want to explain a game that we're going to play during tonight's event. It's bingo. The boards contain words that our speakers may or may not say during their talks. And each time you hear a word, you can mark the square. And if you get 5 in a row in any direction, you win. So to create a board, I'm going to drop a link into the chat here. And so you just want to click on that link to the Google Sheet that I've pasted in the chat. And you're going to create a unique board by making a copy of that sheet. So do File, make a copy. If you don't see a 5x5 board in your copy right away, just wait another minute or click refresh. Sometimes it takes a little time to sort itself out, but it should solve the problem within a minute. And you'll see a 5x5 bingo board with a bunch of words on it. When you hear a word, you can mark the square by highlighting, strike through, bold, whatever you want to do. And then again, if you get 5 in a row in any direction, you win. You can just take a screenshot of your board or download the file and email it to newyork@tasteofscience.org along with your mailing address. And we're going to mail you a prize. You can also announce your bingo in the chat if you want. That would be super fun. Note that once you've started, though, if you refresh the Google Sheet, it will reset your board. And that would probably be bad news if you actually want to win. So don't do that. All right. Great. So now I also want to note that we would love to take your questions during this event. We're going to allow our speakers their time to talk to you, but please use the Q&A function to enter any questions that you have at any point in time. And after each talk, we'll go through those questions and have a lovely discussion. So our first speaker tonight is Erika DeBenedictis. Erika is a former astronomer, recovering computer scientist, and currently a synthetic biologist. During her PhD at MIT Biological Engineering, she studied the origins of life and developed robotic systems for engineering bacteria. Erika recently launched the 2022 Bioautomation Challenge, a grant program that gives researchers access to cloud laboratories in order to improve reproducibility, scalability, and shareability of life science research with programmable experiments. Thank you for being here, Erika.
Thank you for inviting me. This is such a fun event. I'm dying to see what the bingo card entries are that you think I might say, so I'll take a look at that later. Let me just share my screen. All right. So I'm Erika DeBenedictis. Thanks for inviting me. This is really fun. I would love to sort of tell you about my experience with science and life science in particular, and how the types of computer models we're using are changing because of all these advances that are happening in machine learning. And like Catherine said, I actually started my career in science as a computational physicist. So back in undergrad, I did a bunch of astronomy, and I actually worked at NASA doing space mission design, which is where you sort of scope out how to just take metric tons of stuff and schlep it around the solar system in order to support possible missions. And that research area was really attractive to me as a young scientist, because it turns out that with very simple physics, you can actually do a really good job of simulating how to get a metric ton of stuff from here to Jupiter. And so this was a incredibly cool research area that was really accessible from a modeling standpoint. We have really good physics models for modeling how stuff moves around our solar system. And so one day I was like, okay, computational physics is so amazing. I'm going to try using computational physics for something else. And I went and I did a internship at DE Shaw Research in New York City, where they attempt to simulate biology using computational physics. And in particular, they're focused on proteins, which are these magical little molecular machines that do all of the heavy lifting chemically in living organisms. And what astonished me about this problem is that this is so hard. We can simulate with incredible accuracy, like, how our solar system operates, but we can't figure out what happens when you put a protein in water for the life of us, right? Like the bottom up physics first approach to making predictive models in biology works really, really badly. It's so hard. And that observation was what attracted me to biology and what made me really interested in this question of where does real data come from, right? If we make a simulation, how do we know if it's right? And so I promptly went off and did a PhD at MIT Biological Engineering, which was all about taking real, experimental measurements of proteins so that we would actually know whether our simulations are correct. And kind of my perspective on all of this now is that kind of in general, we have this desire to use math and use computers to help us navigate the world and navigate science. And in biology, the particular modeling approach of going from first principles to a prediction is like really hard. It's like really hard going. And instead, there's this new approach, which is throwing the first principles out the window, forget the basic physics, and instead let's start one layer up with some experimental data, and let's build models from the data and go from there. And that's the machine learning approach that is increasingly becoming prevalent because it works really well, it turns out, sort of with biology. And so sort of in that theme, today I'm going to tell you 3 stories. So the first story is the story of AlphaFold 2, which is this algorithm that DeepMind developed that is one of the first really awesome examples of a machine learning algorithm that does really amazing predictions in biology. The second problem I'm going to tell you about was my attempt to solve the next big problem, and how I was foiled 3 times in solving the next big problem. And finally, I'll tell you a story. I'll tell you a third story about something I'm doing now, the bioautomation challenge, that is designed to remove all those barriers that I experienced. Okay, so the story of AlphaFold 2 actually starts much earlier than a couple years ago, and it starts with the creation of the protein data bank. So 50 years ago, when biologists first figured out we could sort of sequence organisms and discover how the ancestry of all the organisms in the big tree of life, they also realized that genomes, DNA, one of the big things that genomes do is they encode the instructions for making proteins, which are these amazing molecular machines that do everything from replicate your DNA to, there's proteins in your eyes that sense light, and every other, amazing chemical activity in your body is mediated by proteins. And it became obvious that if we wanted to understand how biology works, we need to know about proteins. We need to understand how and why they function. And a good place to start doing that is to figure out just what they look like, right? Like, how are they shaped? And people developed this technique called crystallography, which allows you to take a protein and take a picture of it, essentially, figure out what it looks like. And it turns out, so far so good, right? So we know we need to know the structure of proteins, and we have a technique for doing so. Unfortunately, the con is that getting a crystal structure of a single protein can sometimes take years, depending on what protein it is. And so these structures, the structure data for proteins was sort of well known to be really important and really hard to get. And for that reason, people started collecting all the structures that everyone was collecting and putting them in this sort of standardized format called the protein data bank, where they could be accessible to other scientists. They could be stored in sort of a standardized format so that valuable data wouldn't be lost or deprecated. And this became a big thing, and everyone sort of acknowledged this was a standard. And the protein data bank today contains like thousands of biologist years worth of data, right? The sum total of like 50 years of this whole field trying to figure out how proteins look. And it is that data set, which is now composed of hundreds of thousands of structures, that enabled a couple years ago DeepMind to come along and create a machine learning model that performs as well as doing an experiment, which is always the dream, right? You know your model is good when you no longer need to go into lab and measure something. And so this is a graph I really like. So in addition to the protein data bank, people established this competition called CASP, which was held every year, or every other year rather. And it was designed to be a venue where people could sort of benchmark how well their computational models work at doing this sequence to structure prediction of proteins. And so for a long time, there were models that didn't do tremendously well at predicting structure. And then more recently, AlphaFold 1 and then finally AlphaFold 2 came out that do a ton better. And indeed, AlphaFold 2 is sort of spitting distance of being as good as experiments. So it's really, really good. And this success was sort of enabled, I would say, by two key things. The first was having a really well-defined problem, right? Let's predict structures of proteins. And two, having a suitable data set that relates to that problem. And together, that's a recipe for being able to create a predictive model. So now I'm going to tell you about my stab that I have taken at sort of doing the next step here. And so, knowing that the key components are a well-defined problem and a data set, this sort of raises the question, what's the next good example of this in life science? And I and a lot of people have the intuition that probably the next problem that is well suited to be solved sort of with these tools is probably also going to be about proteins. And I think that because we can sort of think about this hierarchy of things that exist in biology, from atoms to chemicals to proteins to cells to tissues to organisms, right? Like all the way up and down. And at the bottom, atoms and chemicals can actually be understood just bottom up from basic physics. And at the top, we have living organisms that can't. And proteins exist in this uncanny valley. It's sort of like the place where things stop being just chemistry and start being kind of alive and unknowable. And so solving problems about proteins is like the easiest of the hard problems, right? It's the easiest of the problems that are actually a real challenge to solve. And in particular, there's one problem in particular that's sort of like the next natural extension of let's predict the structure of a protein, which is let's predict its function, right? At the end of the day, we actually care more about what the protein does rather than sort of cosmetically how it looks. And so this is sort of one step toward sort of engineering goals, one step beyond what we've already done. And so I was really interested in sort of trying to figure out how to approach this problem during my postdoc. And I decided to join David Baker's lab at the University of Washington, which is a lab that does really, really cool protein engineering. So they have developed all of these tools for engineering proteins that have certain functions on the computer. And in particular, as shown on this slide, they have this really cool pipeline for engineering binding proteins. So these are just like little tiny proteins. All they do is just like fit onto another protein, sort of like Lego clicking together. And so their function is binding. And what's cool about the Baker Lab is they've done so much of this engineering, they actually have a lot of data. So they have synthesized and measured more than 2 million proteins at this point. So the proteins are in the fridge, the data is on the file server, and I was like, great, I'm going to get in there, do some machine learning on this data, and it'll be great. And I was, of course, foiled 3 times. So the first time, I get to the Baker lab, and indeed, they do have 2 million data points worth of data. The trouble is, it's really noisy. It was collected using this assay called yeast display, which was never really intended to be quantitative. It was intended to be qualitative, and in fact is. And it was also collected by like 5 different experimentalists over a period of 3 years. And they all did it like a little bit differently because they were all doing slightly different projects and they all wanted it to be a little different. So you can't just go to the file server and take all the data and put it together and learn from it because it's super noisy and really heterogeneous and it just doesn't work. And so this is a common failure mode, actually, where like today, because we don't yet have these computational tools in biology that make the data really useful, we sort of do experiments with the intention of it working today, but we don't think about using the data down the line. And there needs to be like a mindset change where we conduct an experiment both to get a result today, but also with the knowledge that that data may be useful in the future and that we should make it nice. We should make the data nice. So that was the failure mode there. Next, I was like, okay, it's okay. I'm going to just remeasure all the proteins that are in the fridge. So I went on the internet and I looked in the literature and I found this unbelievably cool assay that someone had published where you don't need to know how this works. But basically what they did was they hacked a DNA sequencer in order to take protein function measurements. Coolest thing I had ever seen. And what was really cool about this was it felt like it was the perfect technology to use for my problem. It was something that could measure proteins in amazingly high throughput, millions of proteins at a time. And in principle, it's quantitative. It would give me really nice data. So I went and I tried to get this assay to work. And alas, like many fancy assays, I couldn't get it to work. And in fact, the problem in more detail was that the student who had developed this assay had since graduated, started a company using this technology. And the sort of tacit knowledge of like all the extra stuff you have to do to make it work had been lost because he graduated. He was off doing his own thing. And so this really useful piece of technology was not actually available to me to use. And so here we faced my second time I was thwarted by reproducibility, right? We need to, when someone goes to the trouble to make a really valuable experimental technique, it's worth our while to write down how it works well enough that other people can actually use it, rather than it just being a monument in the literature to their time in grad school. Okay, so I was like, okay, no more fancy, nothing fancy. We're going to go back to the basics. Okay, let's get something just really straightforward and robust. Okay, so I looked around and we actually had a collaborator, Mike Jewett's lab, and his graduate student, Andrew Hunt, who just graduated. Congratulations, Andrew. And they had been using this really robust assay called an AlphaLysa to do measurements of some of our proteins. And so I worked with Andrew to sort of tweak the assay a little bit to make it more suitable for this in particular. And so that's great, right? We can get good data, it's reliable, it works, I can do it, he can do it, everyone can do it. The problem is that if we wanted to conduct this assay and remeasure 1 million proteins, it would cost $1 million. And that's really expensive, right? And if we wanted to lower the cost, we would need access to this fancy robot that we didn't have access to. And so this idea was foiled by the fact that even though we have instruments that allow us to do things in high throughput, most people can't access them. Even people like the Baker Lab is an incredibly well-resourced lab. Even places like the Baker Lab, it takes a really long time, depending on the instrument, to access sort of specialized instruments like this. And so finally, we come to the last section of my talk, in which I tell you a story about how I hope to change all of this, change the status quo, so that all 3 of those problems I just mentioned go away. And this strategy hinges on cloud labs. So cloud labs are this really cool idea, which has been around for some time now, that perhaps kind of like Amazon Web Services, where you farm out computational jobs to some remote place for them to be run, perhaps we could farm out experiments to be run remotely. So you would have a facility, a remote facility that houses a bunch of robotics, which can be scripted. So you can program things to run on the robots. And instead of taking your protocol for an experiment, handing it to a lab tech, having them do God knows what, you take your protocol and you convert it into a script. So you code it up and then you just run it remotely. In principle, this is a really cool way to solve all of those problems. It's a lot more reproducible because you have a script. The data is all electronic anyway, so it's written down somewhere. And people have access to lots of instruments, right? The only trouble is that there's only a couple cloud labs in the US. So there's Emerald Cloud Lab, which is this place. There's also Stratios, formerly Transcriptic. And both of them have focused a lot on industry because it's still kind of a new technology, still kind of expensive. And really the biggest reason research scientists don't use it is because how do you fund it, right? There are no grants that give you access to this. And you can't necessarily just put it in a budget because it's pretty expensive. So I decided to make a grant program. So I launched this program called the Bioautomation Challenge in collaboration with Schmidt Futures and Emerald. And basically how this program works is researchers can submit a proposal for a project they want to do that they think will benefit from being done in this automated setting. They'll get access to the cloud lab and resources necessary to implement that project. And then we'll try to transition them onto their own funding toward the end. And this program, we launched it, I guess, sort of early, maybe in January, accepted applications for a while, and the first awardees are coming online next month. So that's really exciting. And just to sort of tie this back into the previous story I told you, the practice of doing science kind of in this new environment has the potential to address all three of those sort of status quos that foiled my attempts to do a postdoc project. So the whole mindset when you're using Cloud Lab is really different, right? Because a robot is executing your experiment, everything about how that experiment was conducted is written down, and you get a ton of metadata about exactly what happened. And also because all the data is sort of like in one place, it becomes a heck of a lot easier to reference it later, right? You don't have to go running around sending emails to people asking for their data, which was in their Google account that just got deleted. It's just in one place, and you can do a better job of sort of being a good steward of the data. Secondly, once you have a script, it's very easy to share it, right? And that makes it easy to reproduce experiments that other people have done. And finally, this sort of access problem, in principle, cloud labs are this amazing way to democratize access to experimental biology, because as soon as you have a subscription, you have access to like $20 million worth of instruments that most people have no access to. A lot of these are really nice instruments. And so altogether, I hope this effort, as it goes on and future programs we hope to do, will sort of move the needle a little bit, help people get good data sets that cause them to care more about their data, and make a virtuous cycle where people kind of embrace incorporating these learned models in biology. If this is something you found interesting, I also wrote a little essay that you can look up through this little bit.ly link that talks a little bit more about how I see kind of cloud labs playing out in research science. But that's the end of my talk, and happy to take questions.
Awesome. Thank you so much. It's I've talked to you a lot about the Cloud Lab end of things and the Bioautomation Challenge previously, but hadn't heard the beginning part of your story. So that was really cool for me in particular. Yeah. We do have a couple of questions from the attendees. So first off, can anyone access the protein data bank, or do you need like special privileges or anything?
No, you don't. Anyone can access it. You also, now that AlphaFold is out, they have a website where you can look up the predicted structure of any protein. So kind of both of these tools are very accessible, much more so than doing crystallography. I don't even know how to do crystallography, right? So yeah, no, they're very accessible.
Yeah, my understanding is that crystallography involves like lasers and big giant machinery. So yeah, not super accessible.
Yeah, I actually, when I was picking a grad school, I interviewed at a place that does crystallography, and crystallography is so hard. It was one of the first uses of laboratory automation. They have these robots that will try out like all of the different conditions for maybe forming crystals, and then hopefully you get a crystal. So yeah, crystallography is not a fun time, is my understanding.
Cool. Yeah. So next question is, can programs like AlphaFold tell us about complex protein interactions or more just about their static structure?
Yeah, so this is a really good point. So it is a fiction that proteins adopt one structure. Many proteins are functional because they are these dynamic complexes. There are motor proteins, polymerases, like there's tons of variety in terms of like the structures that proteins adopt. I actually kind of want to look up a video to show you. And yeah, so like, you know, one of the simplifications that AlphaFold made in order to succeed at predicting structures is the simplification that proteins just adopt a single structure. And it's not the case. And so I think like there's another direction, which is like, can we predict the structure of proteins that are functional because they have dynamic properties or because they are disordered, right? We increasingly know that the parts of the proteome that can't be crystallized are actually functional. And that's why lots of things work, like transcription factors. And so now that we kind of have the static bits of the proteome down, I think people are really interested in using AlphaFold and other tools to sort of add some of that flexibility and nuance like back into our predictions about protein structure.
Awesome. Yeah, that you actually answered one of the, or you got towards an answer of one of the questions that I was thinking about in thinking about the combination of something like AlphaFold and the work from the Baker lab that you were talking about with the protein binders. I wondered if you were thinking about, in terms of protein function, would it be possible someday to use some kind of predictions based on protein sequences to ask, you know, what would interact with this protein from this sequence based on, you know, its structure prediction and everything else?
Yeah, for sure. That's, I think there are a lot of people trying to do exactly that right now, for sure.
Awesome.
And sorry, this was the video. I don't know if this comes through. This is the DNA holoenzyme. So this is the protein complex that is replicating your DNA like right now. And this is not a static structure, right? This has this crazy thing where it like has all these arms and it has polymerases that are like doing forward and backward replication. And anyway, it's very cool. Proteins aren't static.
Awesome. Yeah, we've got a couple more questions here. So my understanding, and this is, you know, from the audience, not necessarily from me, but my understanding is that AlphaFold does a great job at resolving folding problems that can currently be solved using existing MDS techniques, but that it does a poor job where MDS would fail too. So what classes of molecules remain resistant to structure prediction using AlphaFold or other methods? And then as a second parter, what techniques are being developed to address such molecules specifically? That was a long question. So let me know.
Yeah, no, that's a good one. Yeah, so AlphaFold is good at, so the main thing, AlphaFold kind of cheats. So AlphaFold cheats in the sense that you can infer how proteins are structured by looking at their family tree. And so when you give AlphaFold a sequence, the first thing it does is it like looks up, it builds something called a multiple sequence alignment of that protein with its other family members, which gives it hints about how that protein family is structured. And from there, it's like a much easier to predict like a refined structure. And so it turns out if you give AlphaFold single structures that don't, and like without the multiple sequence alignment, it doesn't work very well at all. And that means that if you have a protein that is like a de novo protein, so something that doesn't occur naturally, AlphaFold's not good at predicting it because it has no hints, right? It has no family tree from which to like infer stuff. So de novo proteins are hard. Obviously, the other category that's hard is like intrinsically disordered proteins, like transcription factors. So some proteins, like you can't predict a fixed structure for them because they literally don't have one. They only adopt a structure when they bind something else like RNA or DNA. And so those are impossible for AlphaFold to handle at the moment because AlphaFold literally doesn't know about DNA, right? Like it doesn't know about any other like molecules. And so it literally can't like predict stuff like that involves like heterocomplexes of different types. And yeah, so lots of techniques are being developed. So let's see. So cryo-EM should definitely be mentioned. So cryo-EM is this sort of the successor, the experimental successor to crystallography that has the capability to allow us to see some amount of protein movement. So that's something that people are really interested in incorporating, building like an AlphaFold that's based on cryo-EM data rather than crystal data, so that we can get some of that flexibility. And then, yeah, just we need a lot more experimental data to start addressing like riboproteins, which is like proteins bound to RNA. We just need like a lot of structures so that we can learn the rules for how these different types of biomolecules interact. But I think in both cases, we face the need to collect a very large mountain of data, and it will not be fast, right? That's the main limiting factor right now.
Gotcha. Yeah, makes sense. I just wanted to ask a few more questions that we have about more of the like cloud lab bioautomation kind of end of things. So what are some things that make measurements different between experimenters?
If only we knew. I mean, yeah, I mean, everything. So like in the yeast display example, everything from like the exact timing, whether you're like a morning person or a night owl, and like whether you let your yeast grow a little too much or a little too little, like each of the days you did the thing. And then on top of that, like, I don't know, did you like, you know, pipette real hard or vortex real hard versus not? Like, what dilution did you do? I mean, just like the details matter, right? Is just the point. And literally just the details matter. Because like, especially when you're working with systems that in yeast display is like maybe an especially good example because it's a experiment that involves as like a key component, like a living thing, like a yeast, right? And so it's even more finicky than like biochemistry, which doesn't involve anything that's actually alive. It's just like, you know, measuring the chemistry of biological things. And so as soon as you have an organism that's actually alive, suddenly a lot of things matter, like timing and temperature and exactly what you put in the media and sort of all of these details just matter a lot. It's really like if you take a yeast out of the fridge today and you grow it up, then you take a yeast out of the fridge tomorrow and grow it up, they won't be the same. That sort of thing. Yeah.
Right. Yeah. I think I vaguely remember another, example of that I read about a couple of years ago where they realized that different experimenters were getting different results with a variety of mouse studies. And the difference ended up being like male versus female scientists were getting different results because the mice reacted differently to the different people, the different genders, which is crazy.
Or there's the counterexample of one time some biologists were purifying some protein they were trying to study. And like the postdoc always got it to work and the grad student could never get it to work. And they just like couldn't figure it out. They were doing exactly the same thing. And then finally they came together and did it side by side together and discovered that what they were trying to purify was part of the circadian cycle, and one of them was a night owl and one of them was a morning person. And so literally doing the experiment at a different time of day gave you a different result, right?
Wow.
Like, yeah, you never know what's up in these things. So you have to be real precise about just like doing everything as much as possible the same.
Great. And just one last question. Do you think there are different types of science or like fields of study that would be more or less amenable to utilizing a cloud lab?
Yeah, for sure. So one of the reasons that I think protein engineering is a good place to start is because it's like right at that junction between biochemistry and like life living things. So it's even with today's cloud lab capabilities, which aren't necessarily everything, you can do most of protein engineering with it. And I think also like because protein engineering, if you test more proteins, it's always better. And so having that brute force is always a benefit in protein engineering, whereas in other disciplines, it's maybe helpful, but it's not like quite so necessary. And so I think protein engineering is a good place to start because you immediately get a benefit from automation and it's pretty automatable, right? And there are many others. I think like microscopy is maybe a good one as well, where that's a field that would really benefit from having all of its data in a standardized place. The instruments are sort of finicky. It'd be nice to house them in a standardized place, stuff like that. So yeah, and I think like part of the bioautomation challenge, we sort of deliberately funded mostly people who do protein stuff and then a few people who do other topics as an attempt to put out some feelers to see like what is the next area that we might want to like encourage to pick up this technique.
Very cool. I hadn't thought about the fact that finicky experiments might be most benefited by a cloud lab because of the standardized approach. That's a really great point.
Yeah.
Cool. Well, I think that's all of the questions that we had, but thank you so much for joining us this evening.
This was fun. Thank you.
It's great to hear your talk. If anyone has additional questions for Erika that they think of later, feel free to drop them in the Q&A and we can add them on. So let me introduce our second speaker, who is Kevin Gardner. He has a Bachelor of Science in Biochemistry from UC Davis and did his PhD in Molecular and Biophysics and Biochemistry at Yale. After a postdoc at the University of Toronto, he started his own lab group at UT Southwestern Med Center in Dallas, and then moved to CUNY in 2013. He also started two companies, Peloton Therapeutics, not the bike company, but a different thing, which he'll tell you all about probably, and Optologics. And these help to commercialize aspects of what he's going to be talking about tonight. So thank you for being here, Kevin.
Thank you, Catherine. Wonderful to be here. Great to get a chance to do an event like this and share a little bit of the work that we have been doing. I share screen. I'm assuming that a fair number of people on the call are U.S. taxpayers, so consider this progress report for all of you who are helping fund the work through your taxes and National Institutes of Health and onwards from there. So Erika gave a great introduction to the field of structural biology, and I am a practicing structural biologist. I actually serve on the advisory board for the protein data bank that she was talking about before, and so our talks I think are going to integrate real well with mine giving a kind of a progress report on how we might use structural biology in the current day and age to go answer interesting biological problems in areas where I think AI has already made an impact and where I hope it will go in the not too distant future. So let's start with our area of biology. I'm really interested in understanding how organisms, individual cells, or something like this mighty fish down here at the bottom middle of the screen, how they know what's going on in the world around them. And I'm someone who is a curious soul, so I don't want to understand that just in one system, but in a whole bunch of systems. For example, how do our cells know if they have enough oxygen or not? How in this 24-hour time-lapse movie, how do these corn seedlings know where blue light is or not? And how does this fish know if it is living in the North Atlantic from Cape Cod up north? It uses a real sensitive sensor for foreign compounds, xenobiotics in the water around it. It's from Cape Cod down to our neck of the woods here in New York. It needs to turn the sensitivity of that sensor way, way down. How are these things all handled? Despite radical differences in us, fish, and corn seedlings. And this is the kind of question my group tackles with structural biology, looking at related but genetically diversified still sensors to light, oxygen, and environmental pollutants. And our tackle here is to use structural biology, and our mantra is pretty simple: structure determines function. If you want to understand how something works, go figure out what it looks like at an atomic level, and then back out of that what evolution did to put those atoms in that order to be able to do a function. If you do that right, we understand how biology works normally, how it's broken in disease, and how we might be able to tweak it or manipulate it, either for novel therapeutic or for biotech purposes. Our playbook is pretty simple. Use the big three kind of founding members of bench science, in my opinion: biology, chemistry, and physics. Biology tell you what questions you should ask. Chemistry gives you some real good intuition about what atoms want to be next to what other atoms and how they're reactive. But then we really need to use the tools of physics to understand what structure looks like at an atomic scale. And this is something that I really want to underscore. Erika introduced a lot of discussion on X-ray crystallography, which is one of three big methods that gets used in 2022 for solving protein structures. One is electron microscopy. There are a class of very high-end electron microscopes called cryo-EMs that are used to take samples of proteins and sometimes with nucleic acids frozen in very, very thin layers of ice and image them, back out from those 2D projections what the molecule was in 3D that gave rise to them. Another is NMR spectroscopy, nuclear magnetic resonance. This is the same physics that MRI uses, just we point our magnets up and down instead of side to side, and we put small samples of purified materials in to go ask in solution what their structures are like and how those can be perturbed. And then the last one that Erika really nicely introduces, X-ray crystallography. I've got to say, this is an experiment that to me sounds like it's on a dare. Take this big ring, which is called a synchrotron, put a bunch of electrons into it that you're moving at near relativistic speed. And as those electrons turn corners, they emit very intense beams of X-rays that we can shine into a crystal, which is a really complicated thing to get sometimes, as she introduced. In the diffraction pattern that comes out, it tells us a lot about what the protein was inside the crystal in the first place. So these are the big three methods, and this has been very productive. Literally, over 150,000 structures are currently in that protein data bank that Erika introduced before. And I want to underscore, all these technologies were not invented for structural biology. We structural biologists are just real clever at using other people's tools to solve our problems. So I want to really underscore that basic research, sometimes in one direction, really benefits other things in ways that you can't necessarily identify on the day that that invention was made. Cool. Thing number one. Thing number two is a lot of the tools you see here were invented by academics, people like us. But there are plenty that were invented by people in industry. The companies that, for example, make or sell a lot of the equipment here have some really bright scientists and engineers at them. They're good. And then government labs have done their share too. So innovation isn't just in one sector or another, but it's all over the place. And then this gets us to talk a little bit about AlphaFold, which Erika did a really nice job introducing. It really serves in three modules. First, you take a protein sequence. Proteins are just linear chains of amino acids. And we run it through comparisons of what other proteins look like that protein sequence amongst all the proteins known in biology. We then take a look at the protein data bank and the 150,000 entries that are there, go in through some special magic secret sauce here in the middle of machine learning. And what comes out on the far side are predicted three-dimensional structures. And critically here, I want to underscore a confidence metric that comes along with it to underscore what aspects of the structure are predicted with high, medium, or low confidence. The protein data bank is about to start serving not only the 150,000 experimental structures, we're going to put 800,000 curated AlphaFold models online later this year, and they're scaling up to 100 million. So soon, people will be able to do a one-stop shop and be able to look at what's been solved by experiment, what's been done by prediction. And I think to be an intelligent consumer of any of that, you really need to have a good understanding as to what level of confidence you should have in any part of a given model. And people are really doing a great job at this. Why do we want to do this? Time, money, and work. Each of these structures that we get experimentally are not cheap in time, effort, and money. And it's also not a guarantee that if we set out to solve the structure of something that that'll work. Expense is huge, right? Imagine a kilometer or two long racetrack where we put electrons in and move them at the speed of light. Not cheap, nor is cryo-EM, nor is NMR spectroscopy. So we are, I think, as an experimental person, I don't view this as competition for my job. I view it as a great tool to help me do my job better, because I want to go in and answer questions about biology and not necessarily solve individual structures each time along the way. AlphaFold in its current iteration, I think it can be as good as experiment in ideal cases. It's not in others, but it's always a good place to go turn to at least develop a model. And hey, what are we as scientists if we're not people who want good models to go beat up on, to go help us design a better experiment to move science forward? All right, so that's the preamble. That's a little bit of how we get these structures, a little bit of why AI is important through AlphaFold. Let's go answer some biological problems, especially in this area of how do cells know what's going on in the world around them. And so our thing here, again, you come into my lab, there are people who work on things out of plants, out of marine bacteria, out of humans, out of mice. Our unifying factor is that all the proteins that are being studied amongst those people all share a little sensory module. You're looking here at experimentally determined structures of parts of these so-called pass domains that are providing the sensing front end to a bunch of different kinds of cellular machinery. The cool thing is nature found these and used them again and again and again to sense oxygen or sense light or sense who knows what. So let's take a look at a couple ways we might put this to action. I'm going to do two vignettes in the remaining part of my talk. Vignette one, how does blue light work? How do plants, bacteria, algae, how do they know what time of day it is? We'll start there. And then we'll go over to a really great biomedical problem, part of the sensors inside of us for oxygen that turned out to be really druggable in an important way for treating certain kinds of cancer. Start with the blue light story first. So, man, I love photobiology. It is my favorite corner of all of biology, because nature has learned to evolve through about 14 or 15 independent classes of photoreceptors, how to look for light everywhere between the UV to the infrared. Holy cow. And to do so sometimes with great sensitivity, right? The human eye can detect a single photon. This is beautiful. Turns out that one class of these that are called the law of domains sense blue light in radically different parts of biology. Those corn seedlings, the movie I showed you before, they're doing it. Bread mold has a circadian clock like us, and it needs to synchronize its circadian clock to time of day. And not just to foil experimentalists, but they need to know what time, I guess, for bread mold, know what time the baker is going to hit the bakery to get ready to eat the tasty bread. So they've got to synchronize their circadian clock to the day-night cycle. Marine bacteria, man, they need to know if the sun's up or not only to know time of day, but also to know if they're living on the surface or deep down in the water column. So they make important decisions like, hey, should I do photosynthesis? Yeah, I got photons. That means I'm shallow, and the sun's up. Let's go to it. Or I'm going to eat whatever carbon I can find around me because I don't see enough sunlight for that. So we got into this a little bit by asking about the land plants. You're looking here at the noble weed, Arabidopsis thaliana. It's a genetic experimental system for our brothers and sisters who are plant biologists. The wild type ones you see here look like they're in a strong wind. That's not wind. That's a scientist went over on the side of these and turned a blue light on it. So they all grew towards the blue light. And if you knock out the photosensor responsible for it, they're blind and they just go straight up. This is phototropism. People have known about this for literally thousands of years. Hey, plants like growing towards the light. And it turns out to be blue light. And it turns out that when you get molecular on it, that there's a kind of an enzyme, a kinase, and then two photosensory domains that we call LOV domains for light, oxygen, voltage, the things that trigger them. So the wiring diagram is like this. Somehow or another, I hit this with light, and it's got to send a signal over to this kinase. Hey, kinase, turn on. And some really bright chemists who were working on this about the time we got into the problem figured out a really important part. Go into one of these LOV domains, go take a look at the molecule that sensitizes it to blue light. It's a flavin. There's some vitamins that we should all be eating or getting in our diet that are this kind of compound, and it sensitizes it to blue light. And we hit it with blue light photon, it ends up going from having non-covalent binding, so there's no bond, no chemical bond holding the flavin into the protein. When you hit with light, a new covalent bond is formed. Holy cow, light is actually doing chemistry on this darn molecule. And as long as that bond is there, some amount of the signal needs to move over to the kinase and say, hey, light's on, activate. When you shut the light off, in a really cool thing, that bond is not super stable. The bond breaks and resets back into the dark state. So my group tackled this using structural biology. We did take some of those NMRs and really wickedly cool lasers that we could shine in through a fiber optic into the top of the NMR magnet. And inside the NMR, we could record spectra like the movie you're seeing here, where literally thousands of times per hour, we're turning the lights on and off on that sample and recording these NMR spectra that give us a readout of the local environment at basically every amino acid in the molecule. Pretty cool. So this is a great example. People were asking a little bit in the Q&A before, hey, proteins are flexible? Heck yeah, that's what we're going on here with all these peaks that are moving around here. It's telling us atom by atom which ones are moving when light is shown on the sample. And we can go make a model out of this. And it turns out in the dark state, we've got kind of the business end of the molecule here. In the spheres, that's the chromophore. That's the part that actually will undergo that photochemistry, bond on or bond off. There's surrounding protein around it. And then there's an alpha helix, a key piece of protein structure that is kind of attached onto the rest of it in the dark. When you hit with light, you generate that bond, and that distorts the protein around it, and off that helix goes, off to the races. Pretty cool. How do you see the light? In this case, nature has evolved a switch to convert something very physical, the energy of an incoming photon, and turn it into something much more biochemical, a protein changing its shape in specific response to that blue light. So how general is this? Well, let's hop in the ocean. And literally, regrettably, we didn't get to. I'm bummed about that. I'm a recreational scuba diver. I would have loved to do this. Instead, I just pick up the phone, open up a computer database. The next thing I see, there are a bunch of interesting protein sequences that are in there from people sequencing bacteria from the ocean. Blue turns out to be a great wavelength of light to look at because it penetrates most deeply into the water column. And it turns out that some marine bacteria figured out how to do some dynamics like this, where now I've got this green part here is the same kind of photosensor like I just showed you. The blue part is actually something that is evolved to bind DNA. And so this is a way that, and you're looking here at a little bit of science fiction. The structure up at the very top, let's see, stop right there. That's an experimental structure solved by X-ray crystallography. The rest of this is just our figment of our imagination as to what's got to happen in this as we go between dark and light, because we think we have an idea about how it goes and binds DNA. So guess what? This is a light-dependent transcription factor. You can go turn genes on and off in bacteria. We're in all kinds of places with this. Well, what do we do with this kind of information? As Catherine mentioned, I set up a couple companies along in my career. One of them was called Optologics, which was to go through and help to design new optogenetic tools. So these are protein photosensors, just like you saw there, but they've been re-engineered to go do something cool with them. What's cool with them? Well, the neuroscientists got onto this first, where they could go take light-controlled proteins, again, from algal sources, and now go overexpress them, say, for example, in specific neurons inside the skull of a rat. And you can then turn those neurons on or off simply by shining blue light on them. As a result, YouTube is now rife with movies of what can happen like this, where in this case, neuroscientists have gone in and had a hypothesis that a certain set of neurons are involved in feeding behavior. So they take this nice rat, they go in and put these optogenetic tools into those specific neurons. And you can see when the light is off on the rat doesn't give a hoot about the food. Light comes back on right about now, and he goes to town. He and the smart food are best friends. He just eat. So kinds of reasons that I'm interested in understanding how you find blue light. It's pretty simple. I'm just wickedly curious about how biology works. But we can take those things that we learn and go turn them into new classes of tools like you see here with the optogenetics. And now the kind of stuff we do have been used literally in hundreds of labs around the world to turn genes on and off. Go express specific genes of your choice simply by shining blue light on either cells up to whole organisms. Pretty cool. All right, I want to finish with the second chapter, getting on over to the drug discovery part, because we do have another area here where AI is really helpful already, and I think is only going to get better. And this is in going and developing not new tools for biotech, but using this information about how we sense what's going on in the world around us to develop new classes of drugs. So in this case, I would like to give everyone a 30-second crash course into the 2019 Nobel Prize, which was awarded to Greg Semenza, Peter Radcliffe, and Bill Kalin, Nobel Prize in Physiology or Medicine, who these three figured out how our cells in a conserved pathway that goes all the way down to fruit flies and worms, how you respond to oxygen, or low oxygen specifically, with a class of proteins known as the hypoxia-inducible factors. What's most important to note is that when O2 is low, you want to assemble this transcription factor called HIF, hypoxia-inducible factor. It's got two components, one called ARNT, hangs out, it's not controlled by O2, but the level of the green subunit is controlled by oxygen normally. In summarizing a Nobel Prize in about 3 sentences, your cells are always making the HIF alpha subunit, but when there's enough O2 around, they should destroy it. And when O2 falls, then you stop that destruction, you let alpha build up, and you go into the nucleus, you go find ARN, and you go control the expression of a couple of hundred genes that helps cells adapt to low oxygen. Hey, let's get more O2 in the blood. Well, let's go make erythropoietin and increase the number of red blood cells that we've got in the blood. Hey, let's go send new blood vessels over to the area that's starved for O2. Well, go express a bunch of angiogenic factors. And all these things over here on glucose metabolism, that's the way cells know how to make energy, make ATP under low O2. So it's a great way to turn it on and off. And in times of injury, like you've hurt yourself, you don't have proper blood flow into an area, you want to turn the HIFs on temporarily. That's cool. The problem is that there are a number of cancers that are caused by the HIFs being out of control and being made all the time, regardless whether O2 is high or low. And we really wanted to put a stop to that. These are horrible cancers of the kidneys, some in the retina, and something that we really wanted to tackle. My group, together chiefly with Rick Bruick's group at UT Southwestern, when we were both in Dallas. So there's a lot of interest in finding ways to shut these off for cancer. By the way, you might also want to turn some of these on, the HIFs on in certain cases for people who have anemia or circulatory diseases, or boo HIFs. If you want to cheat at the Boston Marathon or Tour de France, you might want to turn the HIFs on artificially to increase the red blood cell count in your blood. People have been thrown out Tour de France already for those. Shame on them. Let's focus over on the cancer angle and the inhibitors. So we used X-ray crystallography to solve structures of pieces of the HIFs, and we found a great surprise. Inside of one of the pieces of the HIFs is a big cavity, something that would normally bind some kind of small molecule metabolite, we thought. What exact metabolite that is still a matter of debate amongst us and some of our colleagues in the field. But we knew that it was also the kind of thing that you might be able to find a drug that could go in there and bind to. So how, and a cool part about that is the location of that toehold is right back over with the photoreceptor stuff. And again, I want to underscore, this is a cancer target in us down here in green, Up at the top, this is how plants and bacteria see blue light. And there's a small molecule there to help them see blue light. And here we've got something that tells us something should bind there. Pretty cool that nature has conserved this kind of control in evolution, despite radical differences in the biology in many, many different ways. So let's go find something to be able to go through and bind to this. This is where a lot of the robotics that Erika and Catherine were talking about before are wickedly important. We build libraries of 1,000 compounds and screen them with NMR spectroscopy, for example. So we need robots to build literally 1,000 NMR samples and change them on an NMR. And in a week, we can screen 1,000 compounds for what they hit. If we want to go even a step further, let's go up to high throughput screening. A lot of universities have this. A lot of drug companies have this in spades, libraries on the order of 200,000 to 2 million compounds routinely, and run high-end robotics to repeatedly go through, set up either cell-based or in vitro assays, and monitor which of those compounds are binding. I want to underscore in all these, we're going in blind here with respect to the target. We know that the target does something functionally or structurally. That's what we set up our assays to look at. But we are not really using information on what the target is to help us out. And we found stuff, or even on screens of 1,000 compounds, we saw stuff that started to look good and showed evidence of binding. And then we improved it with what we saw through high throughput screening. And summarizing that work, here's that initial structure, another view of it that I showed you from before. The gray wire is the experimental X-ray structure, and then we've got this cavity in the middle, red showing water molecules in there, the initial structure. And we could then go find small molecule drugs that, guess what, go straight for that cavity, go bind into it. And then critically, again, I really want to underscore this point of proteins needing to be able to flex and have changes in shape to do their function. They can't fit into the pocket as we saw it when there's just water in there. The compound goes in and pushes the protein around it, causing it and reshapes the cavity. And in doing so, pushes the protein outside of that, which pushes on something else, pushes on something else. Next thing you know, the HIFs are shut down. And that was work we did a lot on the academic side of the street, continued over at the startup company, Peloton Therapeutics, and giving rise to a compound we'll talk more about in a minute. This is another place that AI really can and needs to play a critical role here. Given structures of targets like I just showed you from before, it would be nice if we didn't have to go screen 200,000 compounds to go find something, even if AI wasn't perfect, even if it didn't say, hey, this is the drug that you need to go screen, just cut the workload down by tenfold. Yeah, tell us give it 200,000 compounds and tell us who are the 20,000 we should look at. Huge chance for a big savings of time, money, work, reproducibility, all kinds of benefits here. And there are a lot of startup companies that have invested quite highly in this area. So the experiment is being done in one way, shape, or another. And I'm going to be curious to see not only how those turn out on their side, but then also thinking a little bit back to another theme of Erika's talk, how democratized or how accessible will those techniques be to a lab like ours to go chase down the next crazy idea. So in that first case, the crazy idea worked out really well. Peloton Therapeutics made not one, but 2 clinical candidates for compounds that would go into that cavity, reshape it, shut the protein down. Merck bought Peloton in May of 2019, continuing what were then phase 2 clinical trials and beginning phase 3s. And in 2021, this compound, which is now known as belzutifan, was approved for clinical use in the United States for treatment of people who have one of those genetic disorders called von Hippel-Lindau disease, where they're making HIF all the time, regardless of oxygen status. It's about 10,000 patients in the US. Just got approved for similar treatment in the UK about a month and a half ago, with, I believe, a patient population there of about another 3,000 to 4,000. 200,000 people worldwide have von Hippel-Lindau disease. If you had that, you have a 70% chance of developing cancers by the time you turn 60. So this is a game changer of a compound on its own. and is being in extended clinical trials to ask if it can treat other kinds of cancer, specifically the kidney more broadly, and how well it can partner up with some of the awesome drugs that are being used in oncology now, known as checkpoint inhibitors or kinase inhibitors. So with that, this is the summary. I got in this business as structural biologist because I wanted to figure out how proteins work to go do something interesting, like sense what's going on in the world around it. With the help of a great research team, we've been able to answer that in some really different parts of biology. How do plants and bacteria see blue light? And how can an oxygen-dependent transcription factor be controlled by artificial compounds? Put those together, and I think we came up with a really fun story I've been glad to share with you. And I can't wait to see what AI can do to help us accelerate the next round of discoveries from our lab and others. With that, I can't do any of this work without a great team. These are some of the fantastic undergrads, PhD students, and postdocs in my lab at CUNY. Some of the people who have helped pay the bills. And that's what I got. Thank you for your attention. Glad to answer any questions you might have. Thanks.
Thank you so much. They're really cool stories there. We definitely have a couple of questions. First one for me, and I feel like it's kind of a dumb question, but it's something I've always wondered. Why is it always blue light? Like you said that there are sensors for all sorts of wavelengths of light, but I feel like I only hear about blue light. Yeah,
well, that's because your husband has interesting choices and colleagues. So I think that's why you hear about the blue light. The important thing about blue is that the chemistry of the 20 natural amino acids the proteins are made out of are pretty rotten to sense anything in the visible spectrum. You can build in plants use a kind of a okay but not great UV sensor using the amino acid tryptophan. But for anything longer wavelength, blue in through all the colors of the visible into the infrared, you've got to bind a small molecule whose chemical structure allows it to absorb light in the wavelengths of that visible light. It turns out that the general class of molecules known as flavins, flavin mononucleotide, flavin adenine dinucleotide FAD, and riboflavin, that's the vitamin we should all be taking. Turns out their chemical structure of these three ring compounds are perfect for absorbing light around 450 nanometers in the blue. It is all over the place in biology. Every critter uses flavins. So I think it's that ubiquity, of the thing that actually does the tough quantum mechanical part of sensing blue light and turning it into some kind of chemical reaction. The fact that flavins are all over the place means blue light photosensors are very widely distributed around that.
That makes sense.
Great question.
Nature finds something that works and uses it over and over
and over again. You got it.
Cool. Now questions from not me.
Okay.
So one of the three tools you described, cryo-EM, NMR, and X-ray crystallography, much better for understanding what the protein will act like in its natural cellular environment compared to the others?
Yeah, in my opinion, no. I think they all have their strengths and weaknesses in this regard. I do think NMR spectroscopy, I'm biased, it is what I formally trained in. You are doing the experiment there at room temperature in solution. And so I think that helps a lot with what it can give you an answer to. But at the same time, the methods, cryo-EM is done obviously at very low temperatures in usually a thin layer of ice. X-ray crystallography is done by a protein in a crystal. I want to underscore when you say crystals here, everyone thinks of like salt or sugar crystals. Yeah, protein crystals are a little squishier than that. They're at minimum half solvent, but they are still in a crystalline environment. They're oftentimes at low temperature. Liquid nitrogen temp is quite common. But 60 years of experiments have shown that the things that come out of those things at low temp and sometimes in a crystal or in frozen ice actually are pretty decent at telling us what is going on with the protein in solution. Now, a good case where that's really limited is in this bit where I showed you like the science fiction movie of the bacterial sensor that is two, green and blue domain together in the dark, and then they pop apart in the light. Can't do those kinds of gymnastics in a frozen sample or something in a crystal. So their NMR has certain advantages, but there are ways we can, let's say, replicate some of those changes that are compatible with X-ray crystallography and cryo-EM. So to me, each of the three of them has their strengths and weaknesses. No one winner stands out on its own head and shoulders above the other.
Cool, thanks.
Yeah.
Has AI discovered any cellular substructures that scientists didn't already know about?
Not that I'm aware of. And I do want to underscore a bit of this comment that Erika made is really spot on here. The methods where I think AI is really flexing its muscle best in this area are machine learning. And those are cases where I think the results you'll get will only be good as the database you put in. She had a great example of what happens if someone comes up with a fake and artificially synthesized protein that doesn't have something that kind of sort of looks like its sequence in the database. AlphaFold is going to have a real big problem with that. Same story here, at least for now. I like the idea of, as we get continuing advances in high throughput microscopy, both EM and then light microscopy in a variety of ways, I bet there will be cases that as the machine learning methods get better and better, they'll be able to flag yep, I predict it's one of these 10 things, or it's something I can't predict. And I think for now, a lot of those I can't predict answers are not super helpful. They maybe indicate deficiencies in what we've trained the system on more than anything else, or something inside that middle box of neural networks and machine learning. But I bet as those get better, those cases where it says, I can't figure this out, that's less of a problem with the technique and more of a, maybe there's something more here than we had given credit. Looking forward to that.
Yeah, that'll be a really interesting transition point from useless to this is the next question to answer.
Absolutely.
Okay. Awesome. Another question from the attendees. Is the light-driven technology something that can be reasonably used in public health, like changing gene expression in diseases in humans?
Yeah, so I love that. And I will note, I will note, in a prior iteration of this talk, I did it for a series called Science ! Which is here in the city, and it's a mix of scientists and then improv comedians. And my goodness, the improv guys went to town on humans being rewired with optogenetic tools and changing personalities in the middle of a conversation because someone turned the room lights on in the place that they were talking to someone else in. Plenty of fodder there. As for its use in public health, it is already really being pushed in one key area, which is for restoration of vision to some degree in people who have a number of retinal diseases. So the same kind of technology of that channelopsin, that's the name of the protein that got put into the skull of the rat, you can re-engineer that to work in some cells you could imagine putting into someone's retina who's lost vision. If you could couple that into the optic nerve, some very bright people in this area think they've got rats of this, you have the potential of restoring vision for them in certain ways. So that is a light controlled ion channel, just a lot of really important stuff of how it hooks up into the human optic nerve and go from there. But there are reasons to be encouraged in that area. For things like over in controlling gene expression and others, that would be great to imagine. There are a class of drugs currently that are used in chiefly dermatological cases for things that are called photodynamic therapies. So you apply an inactive version of the drug on you, then you just put a light on at the exact region where you want to turn it on or off. You could imagine maybe something with engineered cells that might be able to pull that off as well. But to me, that's way off in the future. The place where maybe I could imagine this coming up in the shorter term are devices. For example, diabetics would like to be able to monitor or have a device inside of them to monitor their insulin levels and in real time go make your own insulin in a device without anyone having to take any kind of measurements or shots or anything like that. And some work by some colleagues of mine in engineering who are using these tools imagine that as a bunch of cells that will make insulin on demand by optogenetic control. So an implantable device, I would bet, aside from that issue with the therapeutic treatment of vision loss, my bet is it'll be, as an implantable device, will be the first place that public health will see optogenetic tools really make an impact in human health.
Very cool. Future is going to be great. Then one last question. So what
Please.
Percentage of binding molecules discovered through screenings actually make it to becoming meaningful drugs?
Yeah, great question. And I think could give an entire talk on that on its own right. So first thing, two kinds of screens I showed you there, one with the NMR and then the one with the high throughput, are actually fundamentally different in that one of them, the NMR experiment, simply was asking whether we could go find a small molecule or not that would bind to our target. If the answer was yes, then we go do follow-up experiments to see if it's functionally important or not. And then the high throughput screen, the one with the 200,000 compounds there, we actually used a second time. You've heard it mentioned, so anyone who's got alpha in their bingo, congratulations. Erika had it for Alpha Lisa or Alpha Liza, and then we used it in a slightly different format. There, the readout was asking whether small molecule binding caused a disruption of two partners to stop interacting, so a functional assay. So in that, the second kind, we knew anything that bound was functional because the way we did the assay. In the first, we just asked for binding, and we got a variety of answers to that. We found compounds that bound and did something functionally interesting. We found some that bound and that were neutral. Those can be interesting as well. And that's good information for someone like us to be able to go to town with. But it isn't, but you raise a really good point, questioner. Binding does not always equal functional change. And oftentimes that means you're not interested in them, but I think in a lot of cases you do. One final thing I'll put a plug out for here. This area of high throughput screening, the NIH really incentivized universities to get into this beginning about 2005. And they set up high throughput centers across the US, et cetera, et cetera. And I would say that there was a lot of investment there that I think we haven't gotten a full return on investment with, because data has been kept out of that in ways that was either not super useful or was useful and no one heard about it. And I think that the challenge that Erika identified of if you are going to go do something, especially at scale, you really need to be asking a bit about how is that data useful for me and my team now? How could it be useful for a fictional future me and my team 10 years from now? But go bigger. How about other people, including people who aren't even really in your community per se? How could it be useful for them 10 years from now? And then how do you get the word out? These are big investments of government and private spending to go make these things happen. It's a real privilege to be able to do those experiments, but I think we always have to look in the mirror and challenge ourselves. How can we make it even more useful than we might otherwise? My 2 cents.
Oh, great points. Well, thank you so much for joining us. It was a great talk.
Thank you.
Just to close out here, I want to say thank you again to both of our speakers, Erika and Kevin, for taking the time out of their evenings to share their work with us. Thank you also to our audience, whether in Zoom or on Facebook Live, and to our New York City Taste of Science team. That's Britta, Vic, Rosemary, Samira, and me, and like all of us working together making this event happen. We hope you enjoyed it. If you have a completed bingo board, don't forget, screenshot it, download it, send it to newyork@tasteofscience.org with your address and get a prize. Woo! We do want to bring you more cool events like this, virtual, in person, hopefully. But with our festival, we lost our biggest source of income. So if you could spare anything at all, we're happy to, take donations. You can just go to that tasteofscience.org webpage and hit the donate button. As a reminder, this event is just one in a series of events that we're holding. This is the second week of our festivals, but there's still more events remaining. We have another one by our team here in New York City that's coming up on Wednesday about how we can build a more sustainable New York City to try to combat the challenges of climate change. It should be really cool. That's Wednesday starting at 7:30. You can, again, go to the website, find the registration link for that. It should be great. We will be posting this video of this event to our YouTube channel, along with the links to all of our guests' Twitters, web pages, so that you can continue to follow their work. And you will also receive a survey of your thoughts about this event. We'd love to hear any feedback you have. There's also information in the survey if you're interested in volunteering for Taste of Science in any city any of our cities across the US. And finally, we really hope to see you in person for events this summer that we're hopefully finally going to be able to put on as this pandemic winds down. So thank you again so much for attending. I hope you had a great evening and learned a lot, and we'll see you at the next event.